Imagine a world without honeybees. Imagine that world without tasty pears, luscious raspberries, or juicy strawberries. Honeybees pollinate 1/3 of the food we eat and provide pollination services with $14.8 billion annually. Unfortunately, honeybee populations are disappearing at an alarming rate. Beginning in 2006, scientists noticed worker bees' disappearance, which led to the colony's inevitable downfall, in what they coined colony collapse disorder, or CCD. One of the many theories behind this tragedy is the widespread use of agricultural toxins and pesticides, which have decreased honeybee health tremendously. Many studies have therefore focused on how honeybees are exposed to pesticides through the pollen and nectar that they collect. My lab, which is Dr. Nye's lab, decided to focus on the bees' exposure to pesticides through water instead. So we asked, why? Why do bees prefer to use agricultural runoff, potentially filled with pesticides, over pure water? So we sought out to determine why bees prefer to collect dirty water and which salt concentrations attract or repel bees. This information will enable us to understand the basic biology of bee salt preferences, will bring to light the unintended consequences of pesticide irrigation practices, and lastly will form the foundation for future studies to help treat colony collapse disorder. So let's head down to the lab and I can show you how we do things. In order to protect us from getting stung, this is the protective gear we need to wear, starting with the full suit, then the gloves, and lastly the boots. But after working with the bees for over a year, this'll do. So we're at the BFS, the Biology Field Station. That's UCSD. I walked that distance. And if you're ever wondering where our beehives are, they're right here. So let's go check out our bees. They're pretty quiet today. Thank God for me. So these are our bees. These are some of them. Oh, I like my camera. This is where I come to capture my bees to do the experiments on. This is my favorite hive. In order to test bees on their salt preference, we captured them at the hive feeder. The bees must be thirsty to search for water in the first place, or else their salt preference won't correlate with their water choice. If random bees were chosen, our results would not accurately reflect our hypothesis. After collecting the desired number of bees, usually about 10 per trial, we put them in the fridge for about 2 minutes just to knock them unconscious. Not too long, or we might kill them. Once the bees are unconscious, they are then placed inside of a small metal rod and taped down to only reveal their upper body parts. Bees' antennas are covered with sense organs, which we use to test what specific concentrations of various salts honeybees prefer. The anesthetized bees are then given 30 minutes total to recover prior to testing, 20 minutes of which is spent in an incubator and 10 minutes in room temperature to adapt. Keep in mind we usually work on 10 bees at a time. I am showing you just one as an example. We then perform a proboscis extension response test on each bee by stimulating both antennae with different levels of salt concentrations simultaneously for 3 seconds. The concentrations are the following: 0%, 0.05%, 0.4%, 0.75%, 1.5%, 6%, and 10%. The salts we used in our experiment are sodium chloride, magnesium chloride, potassium chloride, and disodium phosphate. A 2-minute interval is given in between each concentration to serve as a control on the potential effects of sensitization or habituation of subsequent responses. A response is only counted when the proboscis is fully extended when presented with a drop of each sample. When the experiments are done, the bees are painted red to avoid being used again for future trials. When looking at magnesium chloride and sodium chloride, we found that bees generally have a preference for 1.5%. Honeybees also disliked high concentrations of disodium phosphate and potassium chloride. Interestingly, the primary form of chlorine used to disinfect swimming pools is sodium hypochlorite. There is a strong possibility that this could be a reason why honeybees like drinking from the swimming pool. Future directions of the study include collecting more samples of water sources where bees are collecting their water from. It would also be interesting to identify agricultural sources where bees are frequently collecting water from and adding concentrations of salts bees dislike to see if there is a repelling effect. I hope you've enjoyed our little adventure and learned a little bit more about these cute little creatures. Please consider helping out in our search to find a solution for colony collapse disorder by either donating to organizations or simply changing small habits to save our bees. They need us more than ever.